Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speakers in the chat window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions from the chat window. Your questions in the chat window will only be visible to myself and our speakers. So thank you for joining us today. It is now my pleasure to introduce our speakers, Julie Meneghel and Peter Kilbride from GE Healthcare.
Hello to everyone joining this webinar. We would like to start by briefly giving you some of our background. And as per me, I have been working in the private sector as a cryobiologist for more than 2 years now, after completing a PhD which combined microbiology, biophysics, and cryobiology, trying to characterize and understand the cryopreservation-related injuries in lactic acid bacteria. Today, together with Peter Kilbride, we conduct research projects around cryopreservation, either in-house or through a variety of collaborations with labs and hospitals, mainly across the UK and in Europe, covering the freezing, the cryogenic storage and shipping, and the thawing of mammalian cells.
And I'm Peter Kilbride. I've worked in cryopreservation for 10 years now and have a PhD in cryobiology from University College London. I'm now the Senior Research Scientist at GE Healthcare site in Cambridge and working closely with Julie.
Okay, so we'll start by defining some of the common terms which crop up during cryopreservation discussions. The first one, controlled cooling, is quite self-explanatory. So controlled cooling is a process by which cells are cooled at a specific, reproducible, and predetermined rate. This is essential for successful cryopreservation, and we'll come on to the physical and biological reasons for this later in this webinar. Of course, cooling at a controlled rate is only the start. It doesn't guarantee that the correct controlled cooling program is being used, and for this, the optimal cooling rate must be used, which we will see is cell-to-cell dependent. Each cell type, and even variations within cells such as going from suspensions to spheroids, has an optimal cooling rate unique to the cell system. For somatic mammalian cell suspensions, this is commonly, but certainly not always, 1 degree a minute. For each cell system, it is important to optimize this cooling rate and then ensure that cryopreservation proceeds in a controlled rate following the cell's optimal cooling rate. And the final term we have is glass transition. This is a key term in cryobiology, although it is quite complicated physical process. However, in a nutshell, the glass transition temperature refers to the point at which all biological and physical interactions cease. In cryobiology, this occurs twice. First, inside the cell, known as the intracellular glass transition, occurring around -50 degrees, and then the extracellular in the extracellular medium, which is the extracellular glass transition, occurring around -120 degrees in DMSO-based cryoprotectant.
So, why do we cryopreserve at a controlled rate? Well, it all boils down to cell membrane permeability and water. When ice forms in a biological system, not all the molecules will form ice crystals. In fact, only pure water molecules form ice. Everything else, that is sugars, salts, cryoprotectants, other solutes, and the cells, become concentrated in channels between the ice crystals. These channels become more concentrated at lower temperatures. Cells, being subject to this high solute concentration, start to dehydrate in order to remain in osmotic equilibrium with the solution. This is a good thing because too much water inside the cell during cooling is bad. If cells contain too much water, then ice will form inside the cell during cooling, damaging its membrane and may be lethal. This is exactly what happens when cells are cooled too quickly. If cells are cooled too quickly, the cell does not have enough time to dehydrate, and so this lethal intracellular ice forms. Conversely, if a cell is cooled too slowly, another problem emerges. This high solute concentration on cooling can become increasingly toxic at extended times at relatively high sub-zero temperatures. Cooling too slowly risks toxicity damage to cells. However, mammalian cells are typically robust to this toxicity at sub-zero temperatures. So, as a general rule, if in doubt, going too quickly tends to be worse than going too slowly. Of course, the exact optimal cooling rate will vary depending on each cell's membrane permeability, and so dehydration rate at low temperatures, as well as their robustness to higher cryoprotectant and salt concentrations, which will vary from cell system to cell system. For instance, for T cells, the optimal cooling rate is around 1 degree a minute. It should also be noted that this is the dominant hypothesis for cell death at the moment. However, other factors, including cold shock, membrane oxidization, mechanical ice damage, ATP depletion, and many more factors can contribute to cell damage during the cryopreservation process.
So to carry on with the importance of cooling rate, when optimizing a cooling rate for your cells, an idealized graph will look something like the one shown on this slide. At very slow and rapid rates, the post-thaw recovery is reduced. In mammalian somatic cell suspensions, optimal rates are around 1 degree a minute, while 10 degrees a minute is usually too fast. Cooling as low as 0.1 degree a minute is often acceptable, however, although it does take a long time, but is particularly advantageous for organoid and tissue preservation, especially tissue-engineered constructs used in regenerative medicine.
Now we've covered some of the science, how do you go about optimizing your own protocol? Firstly, we would advise deciding the starting temperature of your process. This should be as low as possible, but above the freezing point of the solution. Normally, the cryoprotectant is added to the cells, and then the cooling process started no higher than about 4 degrees. It's important when starting the cryopreservation process to make sure the controlled rate freezer is at 4 degrees before you begin. The cooling rate will vary depending on the cells, and it's imperative to optimize this for each cell type of interest. However, 1 degree a minute is a good starting point to optimize the cooling rate if you can find no papers in the literature for the cell type you're interested in. You might also have come across some protocols that have something called an ice nucleation plunge step to start the ice formation process. Typically, we would not recommend these as they've never really been shown to be effective. However, for large volumes, say vials or bags greater than around 20 mL fill, having a hold around -30 or -35 degrees to allow for bulk ice formation is also common, and that's shown in the thermal profile presented in this slide, corresponding to a freezing of 8 cryobags filled with about 70 mL each. This hold gives time for the large latent heat of fusion of ice to be dissipated. Finally, the temperature at which it is safe to stop controlled cooling and transfer to long-term storage must be chosen, and we'll discuss this more in the next slide. It's important to emphasize that all groups must develop their own cooling rates. Controlled rate cooling is only effective when the correct optimized protocol is used.
Now, let's see at what point it is safe to stop the controlled cooling and transfer to long-term storage. Well, this is actually a problem that puzzled us for a while, as there didn't seem any consistency in the literature. Some groups, particularly with ovarian cells and eggs, would transfer to long-term storage below around -40 to -50 degrees. Other groups would cool until -80 degrees, especially those using passive coolers. Other groups considered that samples must be cooled below their extracellular glass transition temperature, around -120 degrees, before long-term storage. So we studied the literature, but couldn't find much firm data on this key point. -80 degrees seem to have been chosen as historically, when mechanical freezers were first introduced, the -80 value was chosen to match the temperature of dry ice, which has commonly been used as a coolant. passive coolers started to be used in these minus 80 degrees freezers, which gave a natural endpoint of minus 80 degrees and which carried over to control rate freezers. These extracellular glass transition temperature around minus 120 degrees also had very little evidence. Certainly, cells must be stored long-term below this temperature, but cells would long have ceased dehydrated, and the results from transfer from minus 80 degrees don't appear any worse than those from minus 120 degrees. So to solve this, we carried out an experimental study where we cooled immortalized T cells in a controlled manner to several endpoint temperatures, ranging from plus 4 degrees down to minus 100 degrees. As you can see on these graphs displaying viable cell counts above and cells metabolic activity below over 72 hours of culture post-thaw, for the different endpoint temperatures tested, we found a sharp drop-off in viability for samples transferred at warmer temperatures than approximately minus 50 degrees, but no additional advantage of controlling the cooling below this point. And since this study in immortalized T cells, we have seen similar results in liver and ovarian cells, and other groups have published in a few other mammalian cell lines, all with similar results. This study was published, and you can find the full paper in the reference on this slide.
So why would this temperature be important? What happens around this threshold of minus 50 degrees? Well, as can be seen in the study, it all goes back to the cell dehydration. As cells dehydrate and cooling, all other intracellular compounds, the proteins, vesicles, mitochondria, become more concentrated within the cell. After extensive dehydration, these internal components are pushed together through lack of space, and the cell can dehydrate no further. This is known as the intracellular glass transition, and cells cannot dehydrate beyond this point. When we measured this transition physically, we found it occurred about minus 47 degrees. This makes sense. Controlled cooling is used to allow cells to dehydrate on cooling. When intracellular glass transition occurs, the cells are maximally dehydrated, so no further advantage of control rate cooling is seen, and is safe to transfer to long-term storage without further control of the cooling rate. This exact point has been measured in a range of cell types. However, we would recommend cooling to about minus 60 degrees at least, just to give a bit of buffer room in case some transient warming occurs during transfer.
And now with this final slide, let's summarize what the key physical points in cryopreservation are. Starting on the left-hand side of this figure, cells will undergo a membrane lipid transition, going from fluid to a rigid phase, but this doesn't seriously impact the cellular outcome as it's reversible on warming. Ice will then form a few degrees below zero, around minus 7 degrees in our experimental conditions. However, this is a volume-dependent figure. At that point, cells will start to dehydrate, and this will continue as the temperature falls further. After controlled optimized cooling to minus 47 degrees, the cells will be maximally dehydrated and can safely be transferred to long-term storage, which must be below minus 120 degrees to ensure long-term stability for DMSO-based systems. Right, so we've now reached the end of this webinar. So thank you for following it. We hope you found this quick overview useful, and we can now start to take any questions you may have.
So the first question is, how long can samples be stored safely in?
So I'll take this one. So once you've cooled cells below minus 120 degrees, cells can really be stored indefinitely at that point. At that point, it's not actually a biological event, it's a physical event. So below minus 120 degrees, everything should be stable indefinitely. And there are studies going up to 20, 25 years showing this is the case. The only danger you may have with your cells below minus 120 degrees is accidental transient warming. So for example, if you've got some cells which are stored beside other cells, you might want to retrieve a sample beside it, and that may transiently warm the other samples. So it's important to make sure when taking samples in and out of your storage that only the ones you want warm up and the other ones stay cold. But presuming they stay below minus 120 degrees, and liquid nitrogen it's normally about minus 190, they can be stored indefinitely up to at least 20 years, and that'd be the longest study that's been done so far.
Okay, and why is there a difference in glass transition temperature for intracellular and extracellular environments?
So they're really quite different events, even though they have roughly the same name. The intracellular one is a biological event almost, whereas the extracellular one is a physical event. So because within the cell membrane, you've got a huge number of vesicles and proteins, they form this thing called a colloidal glass. When all the free water leaves, essentially the only water that's remaining is the water that's sort of bound to the proteins or bound to the membranes. And that means biologically metabolism ceases because you've got no water transfer, nothing can actually happen within the cells, you can have no movement of macromolecules. And therefore that's why that occurs at a large temperature. Outside the cell, it is not as highly solute, you don't have lots of proteins and stuff outside the cell, therefore it's a more physical event until you get down and all molecules stop. But there's much more free water outside the cell, which is the general answer.
How does one verify cell viability at the end of cryopreservation under optimal cooling rate conditions? Are there any in-process testing methods?
Well, you would need to thaw your samples first, and then what we are doing ourselves is thaw the samples and then use some specific dyes. So intracellular dyes usually, such as Hoechst or fluorescent diacetate that would enter the cells, and then use a fluorescent microscope to count the number of cells over the total number of cells.
And an important thing to add to that as well is there is a phenomenon that depends when you test your cells post-thaw. So often immediately after thawing your samples, you'll find that they'll have quite good viability, quite good apparent membrane integrity, but then the cells will start dying off and apoptosis channels that are activated. So it's important if you're thawing, say, a treatment that you want to be working for 24 hours, that you do both a viability or preferably functional test on thaw, but then also 24 hours post-thaw to see if there's any apoptosis occurring within the system.
Okay. Have you seen any differences in bag film used for storing cell therapy products?
So I think this question is about
cryo bags and
Sorry, Julie, I'll let you go.
No, it's just that I didn't hear the question properly. Differences between cryo bags and what?
In cryobag film. So when you're storing products, have you seen any differences in the different film types?
So I think in general, when it comes to what bag type or what vial type you're using, the cells themselves don't really know what bag they're in, so there's not really a major impact on the outcome. The only thing to consider is if you have a larger volume, then you will get a big release of latent heat, and so it's better to have a hold while cooling to allow that latent heat to be released, which perhaps isn't as required within cryovials. And it's also important, we didn't really talk about it on the webinar that much, but talk about ice nucleation. The larger the volume, there tends to be less supercooling, it's called, before ice nucleation, and therefore that tends to be better for cells. So you might see a difference in ice nucleation. It tends to be a higher temperature in larger volumes, and some cell types are sensitive to that, and they find a larger volume, therefore better.
Okay, so we have time for just one more question. There are many more questions than one. So continue to type your questions in, and we will pass those directly to Peter and Julie, and they'll be able to follow up with you individually. But for our final question is, will changing the cryoprotectant viscosity help in the cryopreservation process?
I think changing the viscosity will change the cryopreservation process. So one thing that will do, and you see this quite often in glycerol-based systems, glycerol is an often used cryoprotectant, but is much more viscous than DMSO, is you find that you've got to cool more slowly to maintain those follow the phase diagram. So if you cool too quickly and it's too thick, you'll tend to find the ice doesn't form fully on cooling, and therefore when you start warming, you get an event called recrystallization. So in general, having a lower viscosity on cooling tends to be better because it allows the cells to dehydrate more quickly, and it allows the ice to form more fully as well on cooling. So in general, a lower viscosity is better up until the point the glass condition occurs. However, it is cell-to-cell dependent. Some cells prefer glycerol or sugars compared to DMSO, and in that case, it's best just to optimize the cooling rates and then subsequently the warming rates for those cell types individually.
Okay, well, thank you, Peter and Julie.
Thank you all for joining us.
And thanks to our audience. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.